Hello, my name is Morgan Eikanger, and I'm a PhD student in Dr. Rezvani's lab at the University of South Dakota. Metastasis is a common feature of many colorectal cancer tumors and is largely responsible for the morbidity and mortality of patients with colorectal cancer. There's a clear need to develop safer and more effective targeted therapies that are capable of significantly decreasing the high mortality rates associated with the metastatic form of colorectal cancer. This study focuses on Veratridine, a lipid-soluble alkaloid that's extracted from lilac plants. VTD can transcriptionally increase a ubiquitin-like molecule, UBXN2A, that functions as a tumor suppressor protein in colorectal cancer. In this study, I used a set of in vitro and in vivo experiments to show that VTD-dependent induction of UBXN2A can target metastatic characters of colon cancer cells, including tumor growth, migration, and stem cell populations. We received PDX tumor samples from a collaborator, and I ran Western blot to confirm the inverse relationship between UBXN2A and Richter and Richter's downstream products. You can see that when there is more UBXN2A, there's less Richter, and vice versa. When there's less UBXN2A, there's more Richter. Next, Figure 2 confirms that VTD can indeed elevate UBXN2A at the mRNA and protein level. When treated with VTD, you can see that UBXN2A levels increase. Next, both genetic and pharmacological activation of UBXN2A can suppress colon cancer cell migration, as VTD induction of UBXN2A can suppress the epithelial mesenchymal transition and interfere with cancer cell migration and invasion. First, I'd like to draw your attention to the blue and green lines. The blue line is GFP empty, and the green line is GFP UBXN2A, which shows less migration than the blue line, GFP empty. Now looking at the red line, this is GFP UBXN2A, and these cells were also treated with VTD. You can see that there's even less migration. This Western blot just confirms the endogenous UBXN2A resulting from VTD treatment. Next, I wanted to look at the status of cancer stem cells after Veratridine treatment. This is because stem cells are highly responsible for tumor recurrence and drug resistance. You can see that in the SW480 cells, VTD can significantly reduce LGR5, CD133, and CD44 stem cell populations. SW620 is a metastatic colorectal cancer cell line, and it also shows a significant reduction in these populations, but as you can see, it's not as much. So I will need to look into this further. Finally, VTD can inhibit tumor growth in this preclinical mouse model of colorectal cancer. First, I'd like to draw your attention to the red and blue columns. These are both wild-type mice, so they have full UBXN2A gene expression. As you can see, the mice that received VTD have significantly less tumor growth than the mice that received ethanol or the control. Now looking at this third group of mice represented in the green, these are UBXN2A plus minus mice. So they have half the amount of UBXN2A expression as their wild-type counterparts. Looking at panel C, it makes sense that these mice have more tumor growth than the wild-type mice. But interestingly, after VTD treatment, these mice had less tumor growth than the mice that received ethanol, despite having half the amount of expression level of UBXN2A. In conclusion, these findings will provide a therapeutic and mechanistic effects of Veratridine with a significant anti-cancer impact. This will create a new platform for specific mTOR2 inhibitors for patients with colorectal cancer. Thank you, and please let me know if you have any questions.